Multiple kinds of genetic changes can lead to cancer. One genetic change, called a DNA mutation or genetic variant, is a change in the DNA code, like a typo in the sequence of DNA letters. Some variants affect just one DNA letter, called a nucleotide. A nucleotide may be missing, or it may be replaced by another nucleotide. These are called point mutations. For example, around 5% of people with cancer have a point mutation in the Cras gene that replaces the DNA letter G with exit disclaimer. This single letter change creates an abnormal Cras protein that constantly tells cells to grow. Cancer-causing genetic changes can also occur when segments of DNA, sometimes very large ones, are rearranged, deleted, or copied. These are called chromosomal rearrangements. For example, most chronic myelogenous leukemias are caused by a chromosomal rearrangement that places part of the BCR gene next to the ABL gene. This rearrangement creates an abnormal protein, called BCR-ABL, that makes leukemia cells grow out of control. Some cancer-causing DNA changes occur outside genes, in sections of DNA that act like on or off switches for nearby genes. For example, some brain cancer cells have multiple copies of on switches next to genes that drive cell growth. Other DNA changes, known as epigenetic changes, can also cause cancer. Unlike genetic variants, epigenetic changes may be reversible and they don't affect the DNA code. Instead, epigenetic changes affect how DNA is packed into the nucleus. By changing how DNA is packaged, epigenetic changes can alter how much protein a gene makes. Some substances and chemicals in the environment that cause genetic changes can also cause epigenetic changes, such as tobacco smoke, heavy metals like cadmium, and viruses like Epstein-Barr virus.